Okay, so next up we have Professor Kiman Lakau, who has joined us from NAM, Melbourne. Thank you very much for coming to our workshop. Professor Kiman is a professor in statistical genomics and director of Melbourne Integrative Genomics at the University of Melbourne. She's known for leading the development of mixomics, a widely used tool for multi-omics data integration. And today is going to be speaking about multi-omic integration in agriculture. So thanks, Kiman.
Thank you. Thank you. It's a great pleasure to be back at UQ. Actually, I was young when I did my PhD at INRAE in France. And so the talk of Ben was, reminded me of what I learned during that time, but I wasn't working in genomics back then. I was always working in omics or transcriptomics, proteomics, metabolomics, but I was in those labs working on genomics prediction and so on. And then I did my postdoc at UQ, so before I moved to Melbourne 9 years ago. So usually I was talking to David, a lot of my research has been done in medical research, just because of the history and the data I had available. But I thought for this talk, I would give you some examples on how mixomics has been used in agriculture. Okay, so is this work? Okay, so I lead a team of mostly statisticians and computational biologists. We also have some software developers. We're interested in multidisciplinary research, so how do we apply statistics and computational biological methods to real problems. And you will see that a lot of the methods we have developed are technology agnostic. So this is why our methods work really well, regardless of the omics type we're working on. I'm also leading a center called Melbourne Integrative Genomics, and it's really sitting at the interface between biology, mathematics, and statistics. I'm also part of a center of excellence called Cubic, which is actually from UQ, where we're developing quantum computing algorithm for omics data. And some of the discussion we had about, using AI in genomics was quite interesting because in quantum computing, you try to fit the optimization function directly on the data, so it doesn't actually follow, a very specific formula. And I was just thinking, well, maybe quantum computing could work in that case, except that quantum computing is very limited for very large data sets. So we're working on it with my team. Okay, so to give you a bit of context on, how we see a biological system nowadays, is when I started my PhD, we were still in this, reductionist approach where we would make a hypothesis on one single gene. And as a statistician, it's quite easy to solve because then you just have to perform one single test. It can be ANOVA, linear model, whatever you want. And then we had to adopt this more holistic approach with the advent of omics technologies, where you would measure thousands of molecules. I'm talking about molecules in a broad sense. So it can be a gene, it can be a metabolite, it can be protein, but you measure hundreds of thousands of them. And so there's no clear biological question here, or, a clear statistical hypothesis. And I often show this cartoon and I've been showing it for years now, because it's really the field where we're in now. It's like we have many different types of information from heterogeneous sources. It's very difficult actually to analyze them together. You're already facing computational issues with the size of the data you are dealing with, especially in genomics. And a lot of the statistical methods we use completely break down because the number of individuals is much, much smaller than the number of parameters we're measuring. And so a lot of my work has been working directly with people who generate the data to see whether our results actually make sense, because the statistics results don't necessarily make sense, or we can't really validate it from a statistical perspective. And so we went from a reductionist type of approach where we're measuring the expression, for example, of a gene across a cohort. We went to single omics era where we would measure all possible genes or all possible proteins on a cohort of individuals. And now in multi-omics, we're looking at measuring all these omics together, but on the same cohort as well, trying to understand what are the relationships between the different omics layers as well as within the omics layers. But in order to have this holistic understanding, you really need to reduce the dimension space. And so basically what we're trying to do in multi-omics is trying to work out, okay, what are the genes, the proteins, the metabolites that are correlated together? And can they explain the phenotype of interest? So that I can go back into a more reductionist type of approach where I'm generating new hypotheses that I can validate in the lab or in real context. And there's many different ways you can integrate those layers of information. So today we're going to focus mostly on matrix factorization techniques, but be aware that there are many, many others from Bayesian approaches to network modeling, even very basic approaches, which are multi-step, where basically you analyze each omic data set independently. And once you get the results from each of those omics, then you try to understand what is the correlation between the results that you get. So it's a very basic type of integration. And so we developed this package, Mixomics, which is a toolkit for multivariate analysis. Originally, we developed it for bulk omics data. And we have developed more than 19 methods in the package. They have, each method is designed to answer a very specific question from, you want to do classification, regression, prediction, integrating multiple omics in different contexts. And it's been very successful in the past 18 years since I've developed it. So now we have about 50,000 users a year. So it's a big community also to manage. So we developed a lot of resources around it. And it's also led to a spin-out company called Mixomics Pro. The strength of this package is that we're using matrix factorization that can enable users to visualize the results. So this, I'm not going to talk too much about it, but be aware that that's actually the most useful outputs from those methods, because people can really start to understand what's happening in those models and why, how to interpret a statistical result in a biological sense. So in multivariate analysis, Ben already talked about this, but we're saying, looking at a one gene doesn't make sense. We need to look at several genes at a time or several molecules at a time. So we're going to look at a combination of markers rather than looking at univariate markers. We're going to reduce the dimension of the data, and you will see that makes it much easier to understand. And most importantly, the methods I've been developing are able to integrate multiple sources of biological data. And we're going to talk also about supervised analysis for prediction. So I'm going to first introduce, in a very broad context, principal component analysis. It's one of the major basic multivariate method. And then we move on to PLS for data integration. So PCA, I know you've been using that I assume you've been using that quite a bit in genomics. But the way I see PCA is a dimension reduction technique based on matrix factorization. So you start from a very large data set where you have your individuals in rows and you have your features. So it can be SNP, can be genes, can be metabolites in columns. And this matrix is really big. And so from a statistical perspective, it's really difficult. You can't invert a matrix of that size. And so the best way to try to or to summarize this information is to factorize this matrix. So you factorize it into a set of vectors. They can have different names. In PCA, they're called components and loading vectors. And so you can think of, if you're looking at this individual, you're measuring, the expression of 20,000 genes. And so that individual lives in the dimension of 20,000. And so instead you can say, well, can I actually see that individual in 2 or 3D dimensions? A simple way of summarizing this information is to say, I'm going to calculate the average expression values of all your genes. It's going to give me one value, and that's going to be your summary, your score that you assign to this individual. And then you apply the same rule for all the other individuals. You may say, well, looking at the mean is not enough. I mean, I need to look at other things such as the variance or whatever you want. And so you add another component that's going to complement the information that you haven't explained in the first component. And so Ben has talked already about linear combination and how you assign a weight to each of your SNPs. It's the same here. You assign a weight to each of the genes or the molecules you're measuring. And those weights are stored in what we call a loading vector. And so the loading vector is the same size as your original dimension. And it's going to tell you how important that gene is to explain the major source of variation in your data. And so in matrix factorization, the assumption is that if you extract sufficient components and loading vectors, but not too many, because you still want to reduce the dimension of the data, you can reconstruct your original data matrix back. So we really see those components and loading vectors as a summary of your data sets. And the difference with the type of model that Ben was talking about, it's not a random effect model, it's just a linear model. And that instead of having a phenotype that you're trying to model, here the phenotype is actually your component. Okay, so the component is really summarizing all the information. And then you can get a lot of information out of it because you know already which are the genes or the SNPs that are important to define each of your components. So that's just PCA in a matrix factorization point of view. But the reason why I'm explaining this is because once you understand those concepts, you can generalize it to more complex methods for data integration. So the setting we have in multi-omics is you may now have 2 data sets measured on the same individuals, but you may have, I don't know, gene expression, metabolites. They on the same samples, and I will explain why it's important. And what we're trying to do here is different analysis tasks. So for example, you may want to identify what is a set of genes that is highly correlated with a set of metabolites. You may want to predict one with the other. So you want to predict, for example, from the genes, you want to be able to predict the whole metabolomics matrix. Or you may want to understand what is the correlation structure between all these different data sets. And so we're going to use PLS to do this. And so the way it works is the same as a PCA. So I told you that PCA would decompose this data matrix into a set of components and loading vectors. We do the same with PLS, except that now we have to deal with 2 data sets. And what joins them together is that we put a constraint so that the component of the blue data set is highly correlated with the components of the red data set. What that really means is that we're looking for a very specific linear combination of genes that's going to be correlated with a very specific linear combination of metabolites. And you can also use lasso penalization in those models so that you don't have to handle all the genes and all the metabolites. You can shrink some of those coefficients to 0. So you may end up with, say, 5 genes highly correlated with 10 metabolites. So this is how you do variable selection, and you start to identify the correlation structure between those data sets. And so because it works like PCA, you have a set of components per data set. And so you end up with pairs of components. You have pairs of components, T and U for each data set and for each dimension. And then you also have a pair of loading vectors that correspond to each dimension. And similar to PCA, there's this constraint on the orthogonality of those components. So you don't explain the same thing as you go from one dimension to another. And we do that. So the advantage of those matrix factorization techniques is that then we don't have to hang out too long with those large data sets. We can basically only work on the components and loading vectors. So that's very easy to do, to invert those components and do more complicated statistical computation on it. And we use single value decomposition to do that, so it's very fast. Why you need to have those data sets measured on the same samples is because we want a maximal correlation or covariance between those components. So they need to be the same size. So that's one of the main, I think, constraint of those models, multi-omics models. And then you have various flavors. I'm going to show you a few here today. One is you have, for example, one data set, it can be genomics data, and then you have only one single response, one phenotype. Or you can have one data set and multiple response of the same type. So this is where we enter the realm of multivariate multiple regression. So you're regressing one matrix on the other matrix. So I'm going to talk, I've selected two case studies to illustrate the usage of those methods. The first case study, we're going to look at this kind of regression problem. Whereas the second case study, we're going to look at the integration between multi-omics data sets. And so we, in the field, we worked on different setups. And the first one I'm going to talk about is P integration, so this is represented here, where you have the same omics variables that are measured across multiple studies. So we're going to do that first. And the second is the N. So P integration means omics measured on the same P features. And the N integration is you have multiple omics types that are measured on the same N samples. So this is a case study actually that I've been working on with Quentin he was at INRAE and then he moved to ENVT in Toulouse. So it's the vet school. He was interested in understanding the feed efficiency in meat lambs and understand how the feed intake can be modeled with those omics data sets. So we have two types of responses. We have residual energy intake and average daily energy intake, 255 lambs and 13 omics data sets. And I call them omics in a very broad sense. So you can have genotypes, pedigree, but you can also have microbiota. They had metabolomics and many, many, many different things they measured. And not only that's one of the challenges, and the second challenge is that they had data that they collected over 3 years. And so that was a massive problem. So I just represented it here, with those little blocks. So you have those 3 years of collection, and then you have all those multi-omics. And then you have the response, so REI or ADEI. So the problem is we had a massive year effect. This is just a simple PCA where the color represents the year of the collection of those lambs in different omics. So from, metabolomics to other types of fatty acids and so on. And the color is represented with the year is represented as color. So you can see that, I just show you 4 out of the 13, but we had a massive year effect. And so what we did is we used this variant of the PLS method I showed you before that it has been designed, that we developed to do P integration. So basically, for a given omics data set, they measured across different years. And then we still do this matrix decomposition and so on. So we can do classification or regression. Anyway, it's the same idea as what I showed you before. But I move on to the results that he got. So using MINT, he wanted to predict either ADEI, which is how much energy a lamp consumes. And it's the ADEI is calculated, it's a very simple way of calculating this response based on size, growth, body composition of those lamps. But we also had the other response, which is REI, and it's basically the intake that is not explained by any of those covariates, so the residual intake. And then, so what he did is for every single omics data set, he tried to predict REI or ADEI. And so I kind of summarize a bit because otherwise the table would be too long. But you can see first that ADEI is very, very easy to predict. And that's based on those, well, they call it fixed effect, but they're actually those covariates here, so makes sense. Whereas the residual intake is much harder to predict. So this is the correlation between the predicted and the observed phenotype using cross-validation. And basically the heritability, the REI really reflects the heritability in LAMB in those lines. And so the next question was, okay, so those are just the basic results in terms of prediction on each of those omics data sets. But can we actually combine those omics data sets, and is it going to help? And yes, it does to a certain extent. So we increased the prediction to a bit more, depending on the model. And then we also looked at, so he did, I'm not going into detail, but he did some sort of a meta model of those omics. And then we looked at which are the omics that are actually important for the prediction. So it's represented here. It's called a very important measure of predictors. And basically anything above the vertical one line is the omic data set that is important. And so for the REI, it's a mixture of pedigree, genotypes, but also metabolomics data. So most of the omics actually help. Whereas for ADI, which was already quite high, there's not much you can add to it. And the Rumen data were basically very poor predictors. And then he looked at which are the omics, how correlated, how much information do those omics bring, and is that information correlated? So this is a cross-correlation representation of all these different omics. So when it's blue, it's a low correlation. When it's red, it's a high correlation. So as you know, there's a lot of redundancy in those data sets. So genotypes, pedigree, it's the same. Some of the metabolites, were quite high. But what was interesting is that for the REI, the omics actually are highly complementary. They very lowly correlated together to predict REI, whereas ADEI, they are highly collinear. So I think the take-home message from this is that you can improve with some of the omics. You can improve the prediction of your phenotype if your phenotype is actually quite complex. In this case, it was residuals intake. So that's for case study 1, and I'm just going to take a few minutes for case study 2. So this is a data set that I just pulled out from the literature. They were looking at potato and how does the growth of potato react to different stress treatments from drought to heat and drought, waterlogging, and so on. And they measured many multi-omics data sets on that. So for this, what the authors were interested in is understanding a bit better from a molecular perspective what happens under stress. So it's you more in when you look into multi-omics, the first case study was about prediction. This one is more about what is happening here. Can we explain from a molecular level how this potato react to different stress conditions? And so for this, we used a multi-omics a method called Diablo, which is basically a generalization of PLS for multiple omics datasets. So it's the same as before. You decompose your data matrices into components and loading vectors, and each of the components have to be correlated together. You can also include a response of interest. Here, it's a categorical response. These are the stress conditions of the plants. And then you can do variable selection and so on within that. And so what they did is they looked at the correlation matrix between very specific omics datasets. So I pulled this out from the sub figure in the paper, but basically they look at the hormones, some of the hormones they selected and how correlated they are together. They looked at the transcripts as well and the correlation between them. And then they used Diablo to understand the cross-omics correlation between the hormones and the transcripts. And so what they find is that in the control group, the cross-correlation between hormones and transcripts is actually quite weak, meaning that there's not much happening here in terms of how in control condition, hormones and transcripts are not that correlated. But as you move towards waterlogging or, about heat and drought, this is where you start to see very strong correlation between transcripts and hormones. So that may suggest there's a molecular change happening. And then sometimes those correlation are completely disorganized. So this is the case here at waterlogging or when you have a combination of different stresses. So this is really how people use multi-omics in that context to understand those stress conditions. And then you can do more advanced interpretation. So this is where they basically build a knowledge network where each of those nodes here represents different types of omics variables, and they've colored them according to different pathways of interest. And then they looked at how this pathway would evolve under different conditions or different time points to see, which are the pathways that are activated or not activated, and so on under stress. So I hope this gives you a summary of, how you could approach multi-omics. So the key message is we use PLS methods or multivariate methods to reduce the dimension of the data. And that helps us to do feature selection. So you can actually identify, which transcripts, which hormones are important to describe stress conditions, for example. And then we looked at those different case studies. One was more about prediction of a phenotype, but how you handle different batches or independent studies. And the other one was a classic multi-omics integration, where you're trying to understand the connection between those different omics molecules and how they relate to stress. That's it. Thank you.
Merci beaucoup. That was fantastic. Do we have any questions for Kimon? Thank you.
Now it's on. Yeah. My question is regarding dynamic traits. So when we are integrating one matrix from omics with one matrix for traits, what if there are dynamic traits, and then how should we choose and even with omics, there is quite a lot of variance with those dynamic traits. So I haven't seen a single model that can do a lot of that stuff. I tried doing it with weighted gene co-expression network analysis, which is developed for transcriptomics, but I used it for proteomics. But then
Yeah.
we can try that, and it worked fine. But there's
Yeah.
not a lot of models that do that.
Yeah, so that's the problem. I mean, the cross-sectional study, so single snapshot, is you just measure what you have and you try to model what's happening. Actually, we have now developed a whole series of tensor approaches, a mirror of mixomics, but for longitudinal or time course studies. This is really the key to try to understand the dynamics across time. You need to measure them. And there were a lot of methods lacking in that field. So we developed network modeling methods across time, as well as, how do we integrate also the longitudinal measurements? Yeah.
Thank you so much for an amazing talk. I had a question around integrating multiple omic layers where you do not have the same individuals, but they may come from, for example, like a breeding population. So they would have some degree of genetic relatedness. What methods or approaches would you recommend to be able to integrate gene, of course, like gene expression, SNPs, and maybe even proteomics?
Yeah, it's very tricky. So we call this mosaic integration because you have things that are missing. And I've been at Inhai, we looked into those problems, and if you have your population is clones, you can make those assumptions but you need to check, can I match this individual in proteomics with that individual in transcriptomics? That's just one thing that you have to do. I don't think there's any solution. The other one is imputation, where you impute the one that's missing, but probably you're going to do a lot of overfitting there. Yeah, I don't think there's a practical solution yet.
Okay, probably one down question from me. It's probably related with the first question. So, it is interesting that you already explained about the things that we confused most about how to choose multivariate analysis for the data. One things that being a constraint for me is for the temporal analysis. We know that omics measured as the sampling time, but as the causal development window may be open weeks earlier, how do integration framework handle the temporal mismatch between the molecular signal acted and when it was measured? And the second one is, does the multivariate analysis also addresses these temporal variations? Does this also affect the biological interpretations of the data?
Yeah, I think, it relates a bit to the question, I don't think you can model what you haven't measured, pretty much. But if you have a time course study, we have developed spline models to estimate, when the timing is not borderline, where you can, through splines, you can estimate those values. Yeah.
Do have a couple online, but only if we have time.
I'm happy to
I think
answer.
it's worth it to keep going. We'll just have a little bit of a shorter morning tea break. Yeah, I'm making that call.
Okay. So Oma asked, should the success of a new omic technology be judged by its ability to increase prediction accuracy or increase genetic gain per dollar invested? Well, maybe you can answer that question.
No, you do it.
No, but I don't, because I don't have the background on economy versus usefulness.
There needs to be a balance, or I lean towards genetic gain.
Yeah, I would say, I think my element of answer here is, people sometimes want to measure all the omics, and then you work out that actually some omics are not very useful for the prediction. So, Quentin had 13 omics. It was obviously an exploratory study, and Rumen doesn't work. It doesn't bring anything to the table. So it can be seen a bit as a waste, but you need those exploratory studies to say, okay, which idiomics that I really need. So you can do a pilot where you can start to assess, the predictive ability, so that you don't waste money for a much larger cohort. Joanne says there's sometimes little correlation between multi-omics data, especially transcriptomics and proteomics. Is there any other method that can help better integrate those data sets? Proteomics are extremely difficult, and it's very well known that the correlation is actually quite low between transcriptomics and proteomics. People seem to be very disappointed because they follow this biological dogma saying, well, but it should be correlated. And what I say is that we don't know. We still don't know what's happening here. We don't know if it's our assumption, our knowledge assumption is completely wrong, or if we're missing measurements. Because, the problem with correlation is that you're measuring a direct link between a protein and a gene, but you're not measuring, you may not have measured something else that is actually, creating that correlation. So this is where you may have spurious correlation. So things we haven't measured. And the third thing is the technology, the proteomics technology is not very accurate. It's extremely noisy, extremely sensitive to external conditions. And so that may explain also the lack of the poor correlation you can see. And I don't think it's been solved. It's kind of a common problem that, your correlation is going to be quite low if you look at those omics layers.
Thank you again, Kiman, for joining us all the way from Melbourne.